I'd like to welcome everyone to this installment of the Presidential Lecture Series. Welcome, everyone. Before I introduce tonight's speaker, I first want to thank President Dennison for his generous support of this of the seminar series in general and of this tonight's speaker in particular. And that done, it's my distinct pleasure and really an honor to introduce Dr. Rita Colwell as tonight's speaker. I've known Rita for, well, some decades, I guess we shouldn't talk about years. As you can see, Rita is a distinguished professor at the University of Maryland at College Park and also at Johns Hopkins University Bloomfield School of Public Health. She's also senior advisor and chairman emeritus for Canon US, which is a US Life Sciences, which is a diagnostic imaging company. And Rita began her career by obtaining a Bachelor's of Science degree in bacteriology and a Master's of Science in Genetics from Purdue University, and then going on to obtain her PhD in oceanography at the University of Washington. Her research interests include global infectious diseases and water as a resource, and also general health issues related to water, as you'll see from her presentation tonight. She's currently developing an international network to address emerging infectious diseases and water issues with the objective of providing safe drinking water for both the developed and the developing world. Among her other interests are kindergarten through 12 science and mathematics education, graduate science and engineering education, and increasing participation of women and underrepresented minorities in science and engineering. Rita's had a very prestigious career, including having served as the 11th director of the National Science Foundation from 1998 to 2004, during which time she also co-chaired the Committee on Science of the National Science and Technology Council. Also, before going to NSF, Rita was the president of the University of Maryland Biotechnology Institute and professor of microbiology and biotechnology at the University of Maryland, and also a member of the National Science Board from 1984 to 90. She's a member of many national and international societies, including the National Academy of Science in the US, the Royal Swedish Academy of Sciences, the Royal Society of Canada, the American Academy of Arts and Sciences, and the American Philosophical Society. And in 2006, she was awarded the National Medal of Science by the President of the United States. She's been awarded numerous honorary degrees at a variety of higher education institutions and serves on advisory boards for many national and international government committees and policy organizations and private foundations. She's really a nationally and internationally respected scientist and educator and has co-authored or authored 17 books and more than 700 scientific publications, as well as serving on the editorial boards of a number of scientific journals. She's also something of a filmmaker and produced an award-winning film titled Invisible Seas on the subject of marine microbiology. Tonight she'll present her talk on the topic of oceans, climate, and human health, the cholera paradigm. And with that, I give you Rita Colwell.
Thank you very much, Bill. It's a pleasure to be here and to have an opportunity to speak to you about a subject that I really am very, very deeply interested in and trying very hard to try to bring some attention to the fact that we as human beings live as part of many species on this wonderful blue planet, but that many changes are taking place, and not the least of which is that we're vulnerable to the climate changes and especially to the role of infectious diseases. I'd like to take a view that is let's say, cosmic, if I could, towards the topic, and especially try to emphasize the fact that diseases are transmitted by people and through goods transported. And this is just one example of the fact that about 1 billion international arrivals, tourist arrivals, in the past year have taken place, and we expect this to nearly double by the year 2020. So obviously, this is where all the traffic is going to and from. The lighter the area, the more intense the flight paths. So you can see that people and goods move very, very rapidly in this, the 21st century. But fortunately, knowledge travels almost as fast. And in the case of SARS, for example, we were able, in a matter of months, to sequence the virus to be able to determine the source and to trace it to bats as a reservoir in some of the caves of Asia. So as rapidly as people and goods are transported, so is knowledge. Now I'm going to focus this evening on the case of cholera, and that is because it is an extremely good example of how an infectious disease is very much intertwined with the environment in which it is found. Now, the organism is a bacterium with a single polar flagellum, as you can see from the top slide. And this is in a cross-section of the bacterium attached to the villus of the intestine. And this is a scan, the other is a scanning electron micrograph showing how the organism produces these adhesive fibrils and attaches to surfaces. Now, the disease cholera is not a new one in the sense that even in Sanskrit writings, there is evidence that cholera had taken place more than 1,000 years ago. America, India, and the United States. However, I hope to convince you tonight that it didn't travel from one place to another, but in fact the organism is found naturally occurring in the environment. Now this shows the fact that this is from a woodcut that was published in 1832. It demonstrates very graphically the suddenness of the onset of the disease. That is, you can be well, but within 24 hours, if you've ingested water that has the bacterium in it, you can, if you are not treated, lose your life. If the disease is essentially a very severe vomiting, diarrhea, and the loss of fluid, as much as 16 liters in a 24-hour period. Now, this is again from the textbook showing the epidemics of cholera in 1832, 1848, 1868, and 1873. Now, the association with coastal areas is fairly dramatic, and if you'll note that New York, Montreal, New York, Washington, D.C., New Orleans had up until 1900 massive epidemics of the disease. In fact, Washington, D.C. was known as a miasmic swamp because in 1848, miasma, that is bad odor, bad air, was deemed to be the cause of infectious disease. In Washington, D.C., yellow fever, typhoid fever, cholera, malaria, miasmic swamp. Well, today it's not, well, I think it is still a miasmic swamp, but for different reasons. The disease, cholera, again, when it hit New York City, this is supposed to be a graphic figure of the United States asleep while the specter of cholera comes into New York Harbor. It's a little bit reminiscent, I guess, of some of the events recently. Now, the history of cholera is actually the history of epidemiology, because in 1850, 1848, 1854, there were very serious epidemics of cholera in London. London was a city of about 3 million people, bounded very narrowly in a small area. There were no water distribution systems, no sewage distribution systems. You simply emptied chamber pots out the window into the street. It was a city that was crowded, and the epidemics were ferocious. John Snow was a physician who was known for his discovering anesthetics. In fact, he was the first to use opium, laudanum, and other epidemics. Unfortunately, he tested them on himself, so that he ended up actually an addict and died an early death at the age of 50. Nevertheless, Sir John Snow was, through his work as a physician, was the first to deliver, I think it was the 7th child of Queen Victoria, as the first painless childbirth. But he is known for his epidemiological work. What John Snow did was actually, during the epidemic in London, map each of the deaths that occurred, and these dots are highlighted, and it's called the ghost map. In fact, there's a book that was published last year, written by a journalist, called the ghost map. And it's the map of the deaths that occurred in that 1848 and again in 1854. This is the 1854 epidemic. John Snow was able to be perceptive and see that it was around the Marlborough pump that was where people drew their drinking water, that in fact traced down to the Thames and drew directly from the Thames River, which at the time was essentially a raw sewage pit. So he was able, and I highlight it here so that you can see more clearly, the Broad Street pump, the Marlborough pump, and the fact, I misspoke earlier, it was the Broad Street pump that was the one that was used that had the contaminated water. But even having published these data, the acceptance of bacterial-caused diseases, or that is microorganism or bad water causing the disease, the acceptance wasn't really until 20 or 25 years later. The mythology is that John Snow had the handle of the pump removed and that the epidemic abated. But in actual fact, in tracing the history and going through several of the books written about the cholera epidemics and the John Snow epidemiology, it was that the epidemic tapered off in late September. And that is pretty much the story of the disease, that it's very much a seasonal disease. Now, what's also interesting about cholera is that only about 2% are really severely ill and become hospitalized. 5% a moderate illness, and that requires some physician attendance. 18% are mild, and 75% are asymptomatic. So when I give you some of the data for some of the epidemics that have occurred, the hundreds of thousands, it's really the tip of the iceberg, because those actually infected with perhaps a very mild diarrhea and not needing to go to a physician represents much, much larger numbers, 3/4 more actually. The other aspect of the disease that I'll spend a little bit of time on is that in studies that we did in 1969, 1970, in the Chesapeake Bay, particularly around the mid-Bay area, we isolated vibrios that looked very much like Vibrio cholerae. And after some very extensive analysis, we were able to show that we could isolate Vibrio cholerae from Chesapeake Bay. Our detractors in the early 70s didn't believe that you could really isolate this organism from a part of the United States where there had not been cholera for almost 100 years. And so there was a great deal of disbelief that this organism actually existed in the environment. Now, subsequently, we were able to demonstrate further that the organism had another fascinating characteristic, namely that it could be present, but not necessarily cultured. And so we were able to develop a special stain using an antibody, very specific for the bacteria, but linked to a fluorescent molecule that you could see the bacteria directly under a microscope, a special microscope, a fluorescence, epifluorescent microscope, and that even though you couldn't culture it, the organism was present. And so that led us to the hypothesis that this bacterium lives year-round in estuaries and in coastal waters, and that it goes into a dormant stage between the epidemics. But the interesting thing is that the bacterium in its dormant stage And our detractors again said, well, it's probably dead and not really alive. But we were able to show that the same bacteria, when treated with an antibiotic, nalidixic acid, which causes perforations in the cell membrane, and then with added yeast extract, which the cell could then take up, the cells elongated, and they were able to respire, and they were very much alive. So with the right trigger, they could be then induced to begin to metabolize. And so we concluded that there is this survival point where in previous textbooks of microbiology, it had been that, shown that the growth curve was the beginning of growth, a logarithmic growth phase, and then a kind of a stationary phase, and then decline and death. Well, in fact, these organisms that you can't culture are very much alive and able to be resuscitated under certain conditions. And you can see the bacteria as shown here under a scanning electron micrograph. These are rounded up, sort of dormant stage bacteria. And by electron microscopy, this is an actively growing strain, and this is a strain that has a very much thicker cell wall. Now, what's very interesting is that when we took water from Bangladesh, where the bacterium could be detected and where cholera was taking place, we found that the same situation occurred, namely that the bacteria could undergo this non-culturable stage where after only about 10 or 15 days you couldn't culture it, but you could still detect it by these microscopic techniques that it was very much present. And so this has become a kind of a standard hypothesis of this dormant stage of these bacteria that can be resuscitated by passage through an animal, through using rabbit loops, where then they could be recovered. And in some human volunteer studies, we were able to show the same thing. Now, this was done very carefully, and not by graduate students or my postdocs, but this was done at the University of Maryland Center for Vaccine Development, where they do such trials under very carefully controlled conditions. And there we were able to demonstrate that indeed, after several days, after ingesting these non-culturable strains, from which the toxin gene had been excised genetically, so that these were not toxigenic strains, that you could then culture the organism from the patients. And so we were able to demonstrate in our studies in Bangladesh that indeed these bacteria were very much associated with plankton, and that when you couldn't culture them, they were still very much alive and present on the plankton. This is an example of the kind of plankton with which the bacteria are associated, namely the copepod. This is a female copepod. These are the eggs in the egg case, and the bacteria are very heavily, as you can see here, coating the surface of the copepod. So this led us then to the notion that cholera is really like malaria, transmitted by mosquitoes, by dengue, transmitted by mosquitoes, a vector-borne disease, but in this case, the vector is a copepod, one of the plankton species. And here you can see the large numbers of the Vibrios on the surface with a scanning electron micrograph. So because the bacterium has a very powerful chitinase, an enzyme that breaks down that very rigid structure that is on crab shells and on the surface of copepods, that it plays a role in nature in recycling the carbon that makes up the structure of the organism. So it can never be eradicated because it plays an important role in the environment. And here, with this stain that I told you we had developed to detect the bacteria, you can see that on the surface of the copepod, many of the Vibrios can be found. So this relationship is quite specific. One of the students has shown that when you provide non-cholera Vibrios and cholera Vibrios and present them to carefully washed and prepared different copepods, that there's a specificity for certain copepods, and the pathogenic strain outcompetes the other strains. So this provides us then with this background of the disease. Now, let me take you to where you find cholera. The darker the color, the more intense the epidemics. Notice that Peru, 91, 92, had a massive eruption of cholera, hundreds of thousands of cases, thousands of deaths. Mostly, we have assumed that this disease is associated with the countries of Asia, Malaysia, Indonesia, India, and specifically Bangladesh. And here I take you to where we have done most of our work, the Himalayas. The monsoon rains washed nutrients into the river system. Taking you now to Matlab, small village near Dhaka, the capital city of Bangladesh. The darker the area, the hotspots of cholera. And take you now right to the village itself, and you can see the hotspots within this region. This is more like let's say a province or a county in Bangladesh. The blue is where the ponds have been excavated, and the earth that's taken out to create the ponds for the water provides around the ponds the mounds where houses are built. This is the river system, runs into the Ganges, and this is the hospital where we've done most of our work. Now, typical village, typical home in Bangladesh around one of those ponds. The villagers draw their water for drinking directly from these ponds. One corner is the latrine, another corner is where the washing clothes and utensils is done, and another is where the water is drawn for drinking. Obviously, the bacteria don't behave themselves and stay put, so this is not exactly a sanitary or safe situation. The disease can strike in 24 hours. This victim is being brought to the cholera hospital, shown here, which when I started my work back in 1975 was essentially a cement slab with a canvas roof, and during monsoon rains you simply drop the canvas sides to the hospital. It has now been much more modernized. This is a cholera cot, crude, still used, because it's a mechanism for measuring the loss of fluid very imprecisely but usefully to determine how much must be replaced either through intravenous solution or through oral rehydration. A major discovery was made in India about 2 decades ago that with table salt, bicarbonate of soda, and just table sugar in the right proportions in a liter of water, sipped continuously, replenished the fluid. We now have this marketed in the United States. If you have had young children who have had diarrhea or a vomiting episode and you've gone to the pharmacy to get Pedialyte, that in fact is oral rehydration fluid that was developed in India to combat cholera. The studies that we have done have been in this part of Bangladesh, Bakaganj, just across this is in the Bay of Bengal. India is down in this area, Calcutta is down about here, and then Dhaka, the major city, and then Matlab, which I just showed you, is located about so. So much of our work has been done in this region. It's called the Barrier Islands because during the monsoon rains, some of this becomes heavily flooded. This past year, 2007, in August and September, was a very severe year of flooding due to some of the climate events. And 1,000 patients per day entered the cholera hospital for the entire month of September. So it was one of the bad cholera years. We were able to do a long series of studies for 10 years funded by the National Institutes of Health. And we're able to show that in the spring months, February, March, April, May, June, spring epidemic, and then later, October, this is in 97, 98, and then 99, spring and fall are the peaks, and that has been true for about 100 years, spring and fall, major epidemics. We were able to show by measuring a variety of environmental parameters, salinity, temperature, dissolved solids, dissolved oxygen, conductivity, the weather events, the relationships of all of these factors gave us a predictive model. And the predictive model is that very dark line that you see, the black line, and then the upper limit to our model is a dotted line, and then the bars are the actual cases. So with the predictive model, we were beginning to close in on being able to determine just when and where and how intense those epidemics would be. And we now have, in fact, determined just how this disease is transmitted. In the spring months, there is warming with increased sunshine of the sea surface. There is increased rainfall that occurs in the spring months and again in the monsoon period. By measuring the pH and dissolved oxygen, we have noted what are the parameters that lead to the increase, first in phytoplankton, that is responding directly to sunlight. These are the microscopic plants of the sea. And then that's followed, the phytoplankton bloom is followed by the zooplankton bloom, which is sort of the microscopic cattle of the sea that graze on the plants, the phytoplankton. And because these carry the Vibrio in their gut, just as we carry E. coli, Vibrios are the intestinal flora of the copepod, and on their surface, they become much more numerous, and then each one of them can carry 10,000 to 100,000 individual Vibrio cholerae cells. And it takes about 1 million per teaspoon to cause the disease if you ingest it. And so if you go from a 10 or 20 copepods to 100, you've got essentially an infective dose in that liter of water. And then that creates the disease in the person that drinks the water, and then person-to-person transmission follows. So what's important is the phytoplankton followed by the zooplankton blooms, a model created by John Delaney of the events that occurs in the surfaces of the ocean. It struck us that we could probably measure this by satellite, and this would allow us a mechanism for being able then to determine when these epidemics would take place. And so we were able then to utilize a series of satellites. These monitor different aspects and have different sensors. But by monitoring chlorophyll, sea surface temperature, and sea surface height, we were able to enhance the model that we were building, understanding that the bacterium is enhanced in its growth and its association with plankton, and that then the disease is caused by people drinking the water that have the plankton carrying the bacteria. And sure enough, by doing satellite measurements in 1994, we published this in 1996, we were able to measure sea surface height because the winds drive the Bay of Bengal plankton populations up into the river system, and sea surface temperature. And then by matching sea surface temperature, shown here in blue, we found that it preceded the cholera cases very, very closely. So this gave us then a mechanism for determining when the epidemics would occur. Shown here just more clearly, the spring and fall cholera epidemics preceded by the increase in sea surface temperature, which allowed us to predict then when the cholera epidemics would occur. Now, it's important to understand that the El Nino events, and this is the 97-98 El Nino. Notice May, June, the temperature, the red is the warmer the temperature. This is August, October, and then again November, the temperatures begin to warm in the Bay of Bengal. And then again in 1998, February, March, the warming of the sea surface temperature, as you can see in that El Nino year. And then the second peak occurs in August, begins to occur September, October, November. So this is all satellite imagery. This is from NASA, downloading the data from NASA. So you can see that there's a very distinct pattern that occurs year after year. And by building a very complicated model that took into account satellite imagery, the ground truth measurements we'd taken over a 10-year period, we were able to calculate then very precisely. Now here are the cholera cases. These are actually counting the number of people who've come down ill, shown up at the hospital in Dhaka, Bangladesh, with very severe cholera. Some die and most fortunately survive. And these are data for 98 through 2000. I'd shown you the results for 94, 95, and this is the 98 through 2000. Here are the data for sea surface temperature in green, the sea surface height in blue, and the chlorophyll levels in yellow. And then if you correct for the time lag for the numbers of cholera vibrios to become increased, and then you overlay it, you find how closely this approximated. There's an unusual situation in this particular year because there was a delay, there was a temperature anomaly and a delay in the monsoon rains, and there was no spring peak. So we are able, however, to build this model that allows us to predict the cholera cases. We asked ourselves, if this was valid, and this was in August of 2004, when we were doing the final analyses, if this was valid, then we should be able to predict a few months earlier, just from the satellite data and the ground truth data, what the numbers of cases should be in the earlier 3 months. And so we did the calculations, and then we made our estimate, which was, the estimate was about 24 or so cases per 1,000 of cholera victims coming to the hospital. And then we emailed Bangladesh and said, what were the numbers of cases for June 14, 2004? And the answer we got was that the actual cases were 25 to 26. So we now have a model that allows us to predict when, where, and how intense the epidemics will be. Now, it's all well and good to have used molecular biology, genomics, satellite imagery, high-test types of computation, but what does this do for the villagers? If our hypothesis was correct, then the bacteria that are on the plankton could be removed by simple filtration. So we did some experiments in the laboratory with different kinds of cloth. We used Chinese poplin because that's available in Bangladesh and it's not too expensive. We used the t-shirt material that men have t-shirts made from. And we used sari cloth, that very filmy cotton that women used for their dresses in Bangladesh and India. And we found that if you folded it about 4 or 5 times, it removed 99% of the bacteria because they're attached to particulates and to the plankton. So we were able to convince NIH to fund a 3-year study. Actually, the Nursing Institute. This was too simplistic for the National Institute for Allergy and Infectious Diseases. It wasn't technical enough, so they tossed it over to the Nursing Institute. Bless the Nursing Institute. So they funded us. And we did a 3-year study where we had essentially extension agents, women who went to the villages, and we did a very careful statistical array sampling regime of villagers that would filter and village areas that would not. And then we did a direct comparison, and we had the women go to both villages, the controls and those that filtered, to discuss safe drinking water. But to the villages that would filter, they were instructed on how to filter. That is to take about a yard of old sari cloth, the older the better, and fold it 4 times, and then cover the calash that they used to collect the aluminum pot. They used to collect the water for bringing up to the household for the daily use, but first filter it through the folded sari cloth. We were able to reduce cholera by 50%. by this very simple technique. So this then is a mechanism for taking the kinds of advanced science-based knowledge and provide the benefit in an area where the technology of water purification and delivery probably won't be able to be delivered there for at least another decade or more. And it was pretty obvious to the women that the water that had been filtered didn't have all this cloudy stuff which you could actually see swimming, and which we explained to them that that's what made their children sick. What's very interesting is that two things. One is that one of the reviewers for the proposal to NIH said this is not going to work. Men will never drink water that's been filtered through sari cloth, worn, used sari cloth, unclean, won't happen. So we did a 3-month compliance test, and we found out the men had been using sari cloth anyway to filter flies from their beer. So much for... So the next time your proposal gets rejected, remember that reviewer. The other is that the question was, will they continue to filter? So we did a study last year, which was now 4 years after the completion of the 3-year study. And we found that 60 to 70% are still filtering. The difficulty in determining whether it's effective is that the control villages have discovered this and they're filtering as well. So we're having a difficult time. We have to actually interview each family individually to find out who is really filtering on both sides of the control and the villages. But we do know that overall, in that area, cholera is at its lowest ebb, except for the monsoon period when the floods occur and everything kind of goes to pieces. But overall, throughout the year, cholera is at its lowest incidence. Now, I mentioned the El Nino because in Latin America, in 91, 92, just up till December, there were at least almost 350,000 cases and about 4,000 deaths. And by the time that particular El Nino finished, there were about 500,000 cases. It overwhelmed the facilities of Peru and Ecuador and Colombia and even in Mexico. The deaths, I think, were up into the 10,000 range. So this was a very serious time for that part of the world. Now, also, I should point out that it did not, the data I just gave you, did not include the numbers of cases during that year in Bangladesh. So there were close to 1 million cases worldwide of cholera and maybe 20,000 deaths overall from the disease. And this shows you the trend, the cases in Latin America in 91, 92. Because we had developed a rapid diagnostic kit, we were asked to help, and we went into Ecuador because with this kit, we were able to triage those who had cholera and those who had other infections that did not require a replenishment of fluid lost, because the numbers of intravenous units were very limited, and it was critical to make sure that the cholera patients got the intravenous fluid. Peru had massive outbreaks in Lima, but also in 2 other cities about 1,500 kilometers apart. And we were able to show that the 97, 98 epidemic, El Nino, which I've done fast forward here just to show that this was a massive El Nino, the 97, 98 that influenced the waters, the surface waters of Peru and other countries of Latin America. We were able to, this shows the sea surface temperature, the sea surface, the chlorophyll, and the sea surface height anomaly, that it was a much, much higher height, sea surface waves action and tidal action. What we were able to do in 97, 98 was carry out a really fascinating experiment. It was predicted in 95, by 95, that there was going to be another El Nino, this rapid warming of the temperature in that 97-98 period. So we were able to bring the public health workers from Peru, from Mexico, from Chile, from Colombia, from Brazil and Argentina, Mexico, to Maryland, teach them how to do the direct monoclonal antibody and gene probe detection of the bacteria when they couldn't culture it. And then to begin that sampling of water at the sites where there, in 91, 92, had been the massive outbreaks. And sure enough, in September, October, November of 97, they began to detect increasing numbers of cholera bacteria in the water. And by December, the numbers were high enough that indeed cases began to occur. So we were able to do a kind of experiment in the environment that Mother Nature provided for us by having the previous experience of the 91-92 El Nino and being able to extrapolate that to the 97-98 El Nino and to show this correlation. So here is the red are the cholera cases in Lima, Peru, in August 97 through April 98, and here is the sea surface temperature. So that same correlation that we had observed in the Bay of Bengal, we observe now in the case of South America. And in that 97-98 epidemics, we were able to show these are the cases, the vertical, the bars, and then the lines are the sea surface temperatures for Trujillo, Lima, and Arequipa, 1,500 kilometers each from Lima. There had been a paper published that said, it was really, in 91-92, a ship that came in and dumped its ballast water and then seeded Lima harbor with the cholera bacteria. But we went back to the records that preceded the 91-92 epidemic, and then we knew from the 97-98 studies we had done, but in 6 months earlier, in the records in Trujillo, Lima, Arequipa, there were cases of severe diarrhea coming in. So cholera is a dose-dependent disease. The more bacteria, up to 1 million, the more severe increasingly. This was done about 45 years ago by other investigators using a number of human volunteers. And with just a few bacteria, you might get a very mild diarrhea. As I told you, 75% of the cases don't even show up at the doctor's office or the hospital. But it's when you get a very heavy dose of the bacteria that you get the very serious infection. So very clearly, this is an environmental interaction, people and the environment, the bacteria and nature. And in Mexico, again, for that 92, 93 period, and then they had the outbreak a bit earlier in 97, 98 in in Mexico that particular year. But we were able to correlate for Mexico, the Veracruz region, the relationship of the cholera cases. The dotted lines are in fact the rainfall, and the solid lines, if I've got that right, the red line above is sea surface temperature, and the black line is cholera cases, the solid line, and the dotted lines are the rainfall. So this interaction of weather and environment and the disease for the period of 1990 through 1997 was demonstrated for Mexico. A student working in France, Dr. Guillaume Constantin de Magny, had read our papers, and he did the same analysis for Ghana, Africa. He downloaded the data from the World Health Organization database on the computer, and then for this region of Africa, shown here, Ghana, he was able to obtain the data for rainfall and weather events and downloaded the World Health Organization cholera data and used slightly different analyses, wavelet coherency analysis, but what he was able to show was really the same end result, that off the coast of Africa, the correlation of sea surface temperature, rainfall patterns, and the ground truth measurements with cholera could be correlated. So this then provided us with 3 continents: Latin America, the Indian continent, and the African continent, showing exactly the same interactions for this disease. Now they're having some very, very severe epidemics of cholera in Senegal. And in fact, this is a very lethal form. It's 20% of the victims die. It's about 2% usually, but 20% in this region. And it's occurring, as you can see, the deaths are in red. So the numbers of deaths in blue are the total cases. This is very unusual. So we have been receiving the cultures, and we just got a set last week, and we're going to be doing genomic analysis of the cultures. As you can see, the main cities in blue, the major city, is where most of the epidemics are taking place. Now, I'm summarizing the data for Bangladesh, the data for Calcutta, India, the data for Ghana, and the data for South America, Lima, Peru. The same patterns for each of these regions, which gives us now a very powerful predictive model of this interaction of climate, environment, and human health via this infectious disease. We are now working on being able to predict months in advance, and that is by looking at the areas at the tip of India, knowing that the wave patterns, the current patterns, are going to sweep this concentration, this chlorophyll concentration, up into the Bay of Bengal. That is the way the Kelvin wave current pattern goes. So if we can analyze what's happening 3 or 4 months ahead of time, and these anomalies, that means more than normal patterns of chlorophyll, we should be able then to predict 2 or 3 months ahead of time and provide an early warning system for the disease. And sure enough, we were able to correlate this with the zooplankton peaks that occurred subsequent to that a peak of chlorophyll that had occurred 3 months earlier. So we're now developing this prediction, this early warning system for this disease for where cholera occurs in Bangladesh. And again, to remind you that this is the region of Bangladesh in the Bay of Bengal where we've been doing most of our work, and we have for the last 10 years, and in the process of doing another 5 years, a total of 15 years analysis, sampling in these man-made ponds bi-weekly and very intensively, as well as satellite studies, because we're trying to now determine which species of copepod carries the Vibrio. It's just as we know which species of mosquito carries the malaria parasite, we would like to know which species of this copepod carries the cholera Vibrio. So we've done very extensive analyses for the last 5 years. A team in Bangladesh has actually counted every one of the different species, and all of this is now on the computer, and we're just beginning to analyze the data. We've found out some interesting things so far. Copepods comprise about 50% of the population. And this goes according to what we would expect, because that's an area which is mostly a swamp area, and that it's a mangrove area. And the zooplankton specialists know that in mangrove areas, copepods tend to be dominant, the chitin-carrying forms of the plankton. And so now we're in the process of analyzing all of the species to determine which are more commonly present, especially during the epidemics. And from the diversity study, we've been able to conclude that there are 3 groups that are dominant: the rotifers, the copepods, the cladocerans. Interestingly, these are the ones that have chitin, that white, strong, crab shell-like structure that comprises the body of these organisms. 2 of the genera provided most of the diversity of the copepods, the Cyclops and the Dioptimus. So we're honing in on those now. They all contain chitin in their structure, and they yielded the most interesting pattern that is very similar to that we observed for the epidemics of cholera. It's not as dramatic as the sea surface temperature, but we're beginning now to focus in. This is the overall copepod group, but we're beginning to focus in on the given species to determine which ones are dominant at the time of the epidemics. And we're beginning to get a sense of the patterns that will allow us to track down which is the culprit in carrying the cholera vibrio. Now, these are some of the data that we're just in the process of analyzing to determine which of the groups are the ones that we can determine are the most dominant. Sorry, I've got too many of those data. Let me now turn to genomics just for a moment. And I told some of you this afternoon that we had done some dives on the Alvin to the hydrothermal vent. We were able to determine that there are some very interesting, abundant copepods that are present around these very high temperature eruptions of fluid, and around them we can see the copepods swimming. We collected samples of these copepods, and we isolated bacteria. They appeared to be very similar to the cholera vibrio. Did not carry the toxin gene, but otherwise very similar. And since we had sequenced the Vibrio cholerae and published that about 6 years ago, we were able to compare it then with this deep sea Vibrio DNA and to find that it was very, very similar. In fact, 98% similar in its base composition, but also very similar to some other human pathogens, Vibrios, Vibrio vulnificus and Vibrio perihemolyticus. So we're now doing a very intensive study of about, we hope to sequence a total of 50 Vibrio species or strains of cholera over the next year. We've succeeded so far in sequencing completely about, let me go back. Sorry, we've succeeded in sequencing about 15 Vibrio cholerae that have been isolated from patients, and another 10 that have come from the environment but are not the pathogenic or epidemic strains, and then a very closely related bacterium, Vibrio mimicus, and we're now in the process of analyzing these, and we're able to discover a couple of things so far. One is that there is a geographical distribution, strains that are isolated from Madras and from Bangladesh, from Africa, from an early strain isolated from India. So we're able now to begin to determine the evolution and the geographic adaptation of these various strains of Vibrios, and to be able to get a genomic pattern that tells us a great deal about the genes that are the genes common to the species and genes that are, let's say, extraneous, that can be laterally transferred, gained, or lost, and genes that are associated with the species the strain identification. So right now we're in the process of using a variety of sequencing techniques, both 454 and Celexa, as well as the standard techniques that have been used, Sanger and other sequencing. And we're looking at the mechanism of the evolution of this bacterium and trying to determine just how can we define this species. Now, lest you think that this is a disease that hits only the developing countries and is a historical disease, these are the most recent data that I was able to get downloaded from WHO, and you can see that about 60,000 cases occurred in 2004, and it doesn't even include Bangladesh. If you add the cases from Bangladesh, it doubles the number of cases and the number of deaths. So this is a disease that's very much with us. Now, in the United States, we used to have cholera. We don't now because of the water supply, purification, safe distribution. However, the related vibrios that cause disease in humans, Vibrio parahemolyticus and Vibrio vulnificus, closely related to cholera, do cause disease, and you can see that the numbers of cases shown occurring predominantly in Texas, Florida, mostly from people taking shellfish from areas that are marked as unsafe, but because the oysters grow big and fat in the sewage outfalls, people collect them and some people eat them and come down with Vibrio parahemolyticus. And occasionally there are a few cases of cholera that do occur every year in the Bay Area. But also you can see in California numbers of cases. And then something very unusual is happening in Seattle and Alaska. For the first time since we've been taking records on these Vibrio infections, because the surface water has warmed up sufficiently due to climate factors, climate change, global warming, that for the first time last year, there was actually an outbreak of Vibrio parahemolyticus food poisoning. And though this is a very busy table, it shows that there were, in fact, during that period of time, the last year or so, there were 9 deaths from Vibrios in the United States. As you can see, these are not the cholera Vibrios, but mostly Vibrio vulnificus cases, and again, in the spring months predominantly, and the numbers of other species of Vibrio that cause infections. Now, this is a team with whom I've worked in Bangladesh, and I want to emphasize that to carry out this research, collaboration has to be international, and this is a team of physicians, clinicians, environmental microbiologists, the team of sociologists with whom we've worked, the students from the University of Maryland, and then colleagues from other universities, other countries, and certainly the help of NASA, the NIH, and the team at Esri, the satellite firm located in Redlands, California. They have been an international collaborating group. What's also interesting is that Robert Koch, famous German bacteriologist associated with isolating the cholera Vibrio, though I must say that history tells us it was a medical student, Pacini, an Italian medical student, who first described Vibrio cholerae, but he died of the infection, and it was Robert Koch who subsequently did the more elegant description. But Koch said, "I am not a supporter of the exclusive drinking water theory. I think that the ways in which cholera can be spread in a place are extremely diverse, and that as almost every place has its own peculiar conditions, which must be thoroughly researched out. The measures which are of use for protecting the particular place from the pestilence must correspond to these conditions." So Robert Koch, back in 1884, had a sense that the environment played a major role in the transmission of this disease. So with international collaboration, with the kind of biocomplexity from the molecular to the planetary, data gathering, and through the techniques of data crunching with information technology, we are able to track down and show the very intricate interrelationships of this bacterium with the environment. Now, I've spent a lot of time talking about cholera, but I want you to understand that this is a disease because of the fact that we have this unusual and wonderful epidemiological site in Bangladesh where 45 years of epidemiological data have been gathered and where so much knowledge has been accrued, that it has allowed us to really trace down all of these intricate interconnections, and there's still more to be learned. But it is a paradigm for the diseases like West Nile virus, for dengue, for malaria, for yellow fever, for Lyme disease, and for some of the emerging diseases that we're just beginning to understand, like chikungunya disease, which I can barely pronounce, that has moved from Africa to Mediterranean because of the warming conditions. So the patterns of infectious diseases, firstly, connect so much to the environment. With the climate change, we can see clearly that there will be changes in the patterns of these diseases, and that finally, I hope, we will understand that we as humans are but one species in a very intricate pattern of biological life on this planet, and that we really must work together to protect this wonderful blue planet. It is the only one we have. Thank you.
Well, so I'm sure Dr. Colwell will be happy to answer some questions for folks. We have some time left. So before that, I just would really like to thank her once again for a very interesting and informative talk, and for taking the time out of a very busy schedule to come out here and spend some time with us. So one more round of applause. If you have questions, please step up to the microphone, and if you would, give your name as you're asking.
My name is Friary, and I have a question concerning water purification. I think cholera is a disease which in the northern hemisphere all over the world is treated by adding chlorine to the water supply, and that the massive outbreak of cholera in Peru a few years ago was due to the authorities' failure to chlorinate the water supply. And the question is this: do the Bangladeshi authorities not chlorinate the water supplies there? Is this uneconomic or impractical?
You have to understand that these are villagers. We're working in the remote parts of Bangladesh where there is no piping, there is no sewage treatment. There are simply ponds that they dig out, and the earth that they dig out then becomes piled around the ponds, and that's where they build their houses. And so they draw on this water from the ponds, they call them tanks, and from the rivers directly. So there is, in Dhaka city, there is a distributed water supply with some chlorination, but it's a developing country. One thing that I didn't show that I should have is that another aspect of global warming is sea level rise. That part of Bangladesh that I showed you is going to be permanently, 20% of the country is going to be permanently underwater with 1 meter rise in sea level.
I think that speaks to the matter of transmission of the bacterium from seawater to what should be fresh water. Is there a mechanism for this? People don't drink saline ocean water, but try to drink fresh water. How then is the bacterium, which is concentrated on these plankton, transmitted from seawater to fresh water?
Well, first of all, there is that monsoon wind, and the water is brackish. It's not seawater, it's not fresh water in that part of Bangladesh. But there are also these copepods, plankton, in freshwater systems, and they carry the Vibrios as well. So in my hypothesis, for which I have no evidence but just intuition speaking to me, is that probably these copepods have been trapped in earlier glaciated periods in what we now have lakes and streams that are inland, and that they're related, the species are related to the marine species and the estuarine species. So you have a combination of factors, the freshwater forms plus this estuarine form that is where you have the weather events on the eastern side of India and the Bangladesh Bay of Bengal, where you have severe monsoons.
Thank you.
Certainly.
Related question. So the warming of the oceans is mainly an indicator of what's going on inland on the freshwater supply warming? Is that where there isn't a contamination of ocean water to the
That's
fresh
a very
water?
interesting question. We are now beginning to, because some of the satellites now are getting so sophisticated and so sensitive that we now can really monitor ponds and inland lakes. And so we're now beginning, when they launch these satellites, if they can get the money to do it and not spend it to go to Mars, if they can launch the satellites, we will be able then to monitor just those parameters for the inland waters, and that will be very useful. Yes.
The temperatures have not increased over the last 10 years, so how do you justify global warming when there's no increase in a whole decade?
Well, the El Nino events have been very indicative of the amount of warming that can occur. And so with the El Nino events, which have been getting more intense and more frequent over the historical record, have given us the evidence, because the bacteria respond very sensitively to the temperature change. If the water temperature is below 15 degrees, they generally don't grow. It's when the temperature rises above 15 Celsius that they begin to proliferate. And so when the temperature gets up to 16 or 18 degrees, then you can culture the bacteria readily from the water. So these temperature and salinity are very sensitive indicators for the distribution of these bacteria. I can speak with rather strong authority because of the 20 years of research that we have done in the Chesapeake Bay, the Bay of Bengal, and off the coast of Calcutta.
Thank you.
My name is Ford Johnson, and from listening to your talk, it sounds to me like the problem is insoluble without the introduction of sanitation methods and a combination of education to go along with it. In other words, it's the whole society needs to be uplifted in education and science terms. I wonder if you could comment.
I wish you were Bill Gates, because if an investment were made in providing safe drinking water, we would reduce deaths of children under the ages of 4 and 5 incredibly, significantly. Safe drinking water is probably the most important action we could take, and that includes proper sanitation. No question, combined with education. Absolutely. Thank you.
My name is Beverly Young, and I'm a water engineer, and I've worked in Africa, and I agree exactly with what you said. I have a very strong interest in engaging young women in math and sciences, and I just would like you to, expound on that a little bit, since that's one of your interests as well.
I think if we don't utilize the full talent of all of our population, especially in encouraging them to go into science, math, engineering, as a country, we're going to suffer significantly, adversely, from social disruption, from economic downturn, and from the lack of, the innovations that come with creating new jobs, new companies, new industries, and so forth. It's really very worrisome that the numbers of children going into math and science is going down. And I'll use this example. I now spend a lot of time as advisor to other countries, Ireland, Sweden, Singapore, Japan. In Japan, it's a rather insular country, a very interesting and a lovely place. As we all know, the population is aging. The numbers in Japan as well, going into science and engineering, is going down. And it's a fairly macho society, if I will say. So in talking with the Ministry of Science, this was 2 years ago, I told them that they were really making a serious mistake in that they had this problem of an aging population, declining enrollment, and they had two paths they could follow. One is they could import a whole lot of Koreans and Chinese and Indian scientists and engineers, and this is a very insular society, or they could really integrate women into their science and engineering complex. Well, I was very interested. I told them about a program that we had started at the National Science Foundation called Advance, and I was very interested. The next year that I went to Japan, they proudly introduced me to their first cohort of Japanese Advance Fellowship awardees. So they got the message. So it's very clear that they understand that they've got to bring women into their science and engineering infrastructure if they're going to remain competitive in the 21st century. That's a partial answer.
Mark Grimes, what can be done during the monsoon season?
What can be done during monsoons? I think this is a very tough question because it's a country of wonderful people, highly resilient, but very poor. And I think the infrastructure is the big problem. It is, I think, a matter of building the kind of housing and the kind of housing, for example, that's built in Australia, essentially on stilts. And that's because they have these very heavy rains in the spring and swollen rivers and flooding, but they don't lose all of their structure. And I think in the case of Bangladesh, it's a matter really of resources. Oh, I'm sorry.
Hi, I'm Vicki Watson, and I had a question about the dynamics of chlorophyll and zooplankton, which you've tied primarily to temperature increases, and I wondered to what extent increasing nutrient pollution might play a role in this as well. And also if you've looked at just the bacterial communities that are there to see if there's changes in the bacterial community composition that might also explain some of the changes.
Yeah, very, very good question. A Japanese team has done studies, 20 years ago, showing that the kinds of bacteria associated with phytoplankton are very different for the microbiologists in the group. They're the oxidative gram-negatives like Pseudomonas and what we used to call Flavobacterium. And the bacteria associated with zooplankton are the fermentative Vibrios aeromonas groups. So they're very different groups that are associated. Microbiology is very different for the two groups. The dynamics, the question is about the dynamics of the population shifts. Not quite sure I got that question.
Well, I think you were mainly, predicting the cholera outbreaks in connection with temperature increases, and I just wondered if nutrient dynamics
nutrients,
might
I'm
also
sorry.
be tied to
Yeah.
that, and also if you have these community shifts in the bacteria going on.
Yes, I mentioned the monsoon rains. You notice that the fall peak is much higher. And that's because in the spring, you've got the temperature increasing, sunlight, phytoplankton growing, and then you have a modest increase, relatively small increase in cholera cases. But with the monsoon rains, and then as it subsides, there's a lot of nutrient, and then the epidemic is much, much more severe because the numbers of plankton are much higher. So there is an interaction of nutrient concentration. Yes, sir.
I just want to ask, what would you estimate the cost ratio is of hospitalizing and treating people versus the prevention and sanitation?
Well, the cost, monetarily, it's kind of hard to offer, but I can tell you there would be a whole lot more surviving children, in the order of thousands that would survive if we had proper sanitation. The other factor that's also very important is that as the economy improves and as the education of girls takes place, as it has done in Bangladesh, and as sanitation has improved, but still it's a long way to go, the fertility rate has dropped from about 8 or so down to about 4. And so that's another positive result, because the children survive. And instead of having very large families to ensure that a few survive, when you know that your children will survive, then it is the benefit for everybody, the families, the environment, the nation.
I see there's another question, but just on a related topic, it seems there probably are some very low-cost, low-tech solutions that would help. For example, it strikes me that sand and soil are very good filters. A cubic meter of sand can probably do a lot of percolated water, for example.
You're absolutely right. What has occurred in Bangladesh is a kind of a double whammy. The World Bank invested in deep wells, boreholes as they're called in Bangladesh, to tap groundwater, but they never tested it for metal content. And what they did was introduce another epidemic of arsenic poisoning and cancer. So that is why the filtration technique, going back to using surface water, the idea was surface water was so polluted, if you tap groundwater, you'd have clean water. Well, you got just the opposite. However, an engineer at George Mason University last year won the National Academy of Sciences Engineering Prize for developing a very simple, for $10, you can get a sand, charcoal, gravel filter, and with iron added, you can filter the water and remove the arsenic. I'm now teaming up with him to put a pre-filter of sari cloth so that a family then for $10 would have good, he's shown that it works for 5 years, a filter that would provide safe water in the individual home for drinking. And you're right, it doesn't have to be, that's the problem. There's this notion that it has to be very high tech, very expensive, but if you're in a country where you have to fly in the repairman, I mean, you can't just call him on the phone, but comes in to make these repairs. And if you need to depend on filters that are manufactured elsewhere and take, a week to get there, it doesn't work. The simple is what works. Thank you very much. Oh, another question. Yes.
You haven't mentioned boiling water. Is that effective?
Yes, very simple. In Latin America, they post boil water alerts. In Bangladesh, there is insufficient fuel wood. The women actually collect the cow dung and the goat dung, form patties, dry it in the sun, and they do that every day, and that's the fuel used to cook the meals. So boiling large amounts of water for drinking is out of the question. It comes back to an earlier question, why not sanitation? This is a very, very impoverished society, and the simplest solutions have to be the best. But certainly boiling water, yeah, that's the way to go. Interestingly, someone mentioned, an earlier questioner mentioned the fact that in 91, 92, there was this big flap about the chloramines, carcinogens, forming from chlorinating the water. Well, it was even worse in Lima. In the barrios, water is distributed in large tank trucks, and They didn't like to chlorinate the water because it eroded the tank trucks. So they were literally distributing cholera contaminated water. I tested the water and I was horrified to find out that the water contained the cholera, and they were distributing it to the cholera bacteria and distributing it to the barrios inhabitants and charging them for it as well. Thank you. Good.